So hello, my name is Christoph Stein Thoeringer. I'm professor for infectious diseases and translational microbiome research at the University Clinic of Tübingen. And I'm also member of the board of CMFI. It's all centered about translational microbiome research. So we are doing a lot of research on the question of how the microbiome influences clinical therapies. And our major focus is cancer therapy. How individual microbes or their metabolites as the major effectors of the microbiome influence, for instance, immunotherapies against lymphomas or leukemias when we utilize CAR T cells as a novel therapy in cancer medicine. We are also working on chemotherapies and how microbes, their metabolites, affect the chemotherapies, which is very relevant for GI cancers such as pancreatic cancer or colorectal cancer. Another question is about how the microbiome can be stabilized in clinical conditions when we see a lot of stress to the microbiome. For instance, we know that antibiotics can reduce the diversity, can induce compositional changes in the microbiome, which then again affects the functional output of the microbiome. So this is all done, as I said, in the context of cancer medicine, but we are also working on mouse models and cell culture models. We are working on bacterial anaerobic cultures to also dive deep into mechanisms. That's very important for us because we want to go beyond describing the effects of the microbiome. We want to analyze and investigate mechanisms. As I said, we're working on metabolites, and we know that metabolites are the major effectors of the microbiome. And in this context, we are working, for instance, on microbial metabolites that affect tumor growth in GI cancers, that affect immune cells, which are then important for immunotherapy. So we are a Translational Microbiome Research Lab, and we do all our research in a perpetual dialogue between mouse and man. So we have established in the past several clinical cohorts in pancreatic cancer patients, colorectal cancer patients, lymphoma patients, and these patients usually get some sort of cancer therapies such as adoptive T-cell therapies, mainly CAR T-cell therapies or allogeneic bone marrow transplant in case of lymphomas, or chemotherapies if it comes to pancreatic cancer or colorectal cancer. And here in these methods, we are looking into the microbiome. We sequence the microbiome usually with shotgun metagenome sequencing, and then we apply novel bioinformatic strategies to identify microbial functions, factors that are relevant, for instance, for response to chemotherapy or immunotherapy. In this context, we are also applying novel machine learning algorithms because we think that this is the future to identify key components of the microbiome or their factors to really predict long-term outcomes. Apart from these clinical cohorts, we are studying microbial metabolites in cell culture methods, for instance, in tumor cell cultures. We are utilizing a broad range of microbial culture systems from anaerobic to aerobic systems to build biobanks of microbes that we isolate from human biospecimens, and then look into their effectors in co-culture systems. For instance, we culture Enterococcus and Enterococcus lysates with PBMCs that we isolate from patients to identify their immunological relevance in patient cohorts. We worked on Enterococci in stem cell transplant patients where we found that for the first time that Enterococcus as a major commensal of the gut microbiome can impact the development of graft-versus-host disease. As a successor of allogeneic bone marrow transplant in patients with hematologic malignancies, we have a completely novel field of CAR T-cell therapy, and we recently published another paper showing that the gut microbiome and alterations of the gut microbiome through antibiotics, for instance, is a relevant factor for predicting long-term outcomes. This has also been done in international clinical cohorts. If it comes to individual pathogens, we have a large study published just recently on the effects of these pathogens in the biliary tract and the upper GI tract of pancreatic cancer patients, showing that Enterococcus can also be a predictor for surgical site infections and other adverse events that occur after pancreatic cancer surgery. Meanwhile, we are also working on mechanistic models where we, for instance, studied individual metabolites that are derived from gut microbes, and found, for instance, that specific short-chain fatty acids that are produced by commensals upon fiber diets and other nutritive factors that are relevant for shaping immune cell functions in patients, and especially again in CAR T-cell patients. We have also several research results at the moment that show that specific diets or nutrients can stabilize gut ecologies in vitro. Here we, for instance, identified inulin or galacto-oligosaccharides to be relevant for stabilizing complex communities in vitro upon antibiotic exposures, which then again prevents the expansion of Enterococcus and other pathobionts in vitro. If it comes to mechanistic models, we found, for instance, that upon antibiotic depletion of the gut microbiome in patient cohorts, we lose beneficial microbial metabolites, such as bile acids or short-chain fatty acids. And this can be a surrogate marker for adverse outcomes in cancer immunotherapies. But on the other hand, we worked on the mechanism. And here we, for instance, found that specific receptors that are located on the surface of T cells can recognize short-chain fatty acids. And if these receptors are lacking, the T cells undergo apoptosis. They are metabolically unstable, and therefore they are not acting against tumor cells. And in an unpublished paper, we, for instance, showed that short-chain fatty acids, when we give them to tumor cell co-cultures with CAR T cells, the CAR T cells maintain their long-term stability and are able to attack the tumor cells much more efficiently in the presence of short-chain fatty acids. And we have the hypothesis that this is totally dependent on receptors on the surface of T cells and their downstream metabolic efficacies. Over the past decade, we and others have shown that the microbiome is highly relevant for clinical medicine. And this spans the whole range from infectious diseases to neurological disorders to metabolic disorders up to cancer medicine. And we have seen relevance in terms of biomarkers that stratify patient cohorts to help to predict, patient outcomes, starting from immunotherapies to antibiotic administrations up to the level of neuropsychiatric interventions. And we've also seen that microbiota interventions, for instance, on the level of fecal microbiota transfers or probiotics, can be extremely helpful to improve quality of life, improve relapse of infections, for instance, C. diff infections, or even if it comes to cancer medicine, improve the efficacy of immunotherapies. So all our research is driven by a mission to improve wealth and health of our patients. We're working on the microbiome and microbial effectors to improve the outcomes of medical, cancer, and immunotherapies. And if it comes to the future perspective, where do we see our microbiome research going to? We have the vision to provide microbial or microbiota-based interventions to improve the outcomes of cancer therapies. In this context, we're working on clinical trials to stabilize the microbiome through diets. So because we know now already, and we have plenty of research data underlying this concept, that upon antibiotic stress of the microbiome, we need to feed our microbes well. And we think that providing nutrients, for instance, through resistant potato starches or other fibers, we can stabilize the microbiome. And this concept and this hypothesis is now and will be brought to the clinics in high-risk cancer patient cohorts, but also cohorts of patients with infectious diseases, where we try to intervene with the adverse effect of antibiotics and other drugs in these patient cohorts to stabilize the microbiome through diet interventions. So we see and we work on the broad range of biomarkers, microbiome interventions in clinical cohorts, and hope that over the next years we can bring this to the clinics and bring it also to some sort of standard of care. It's a long way to go, but we are fully driven by our mission to really utilize the power and all the sources of the microbiome to make this happen.